we start. Thanks, Jenny, for coming here and give your presentation, and thanks to everybody who is attending online. I guess this is being recorded, so I leave it to you. About half an hour, 40 minutes, and then we'll have some chats. Just to be sure the people here in person will be able to ask questions. Online, please use the chat and I will ask the questions for you. Brilliant.
Sounds good. Thank you, Lorenzo. Let me just get rid of that. Well, thank you for the invitation to be here today. I'm going to tell you a little bit about what my lab does, and mostly a little bit about what the brain does all of the time, because almost all of us see and hear. So how does vision influence listening is one of the things that happens in my lab. We also look at auditory spatial coding and auditory scene analysis, so how your brain takes the sum of sounds that arrive at your ear and actually separates them out into sources that you can recognize and understand. And a lot of our work uses, or some of our work uses human listeners, and a lot of our work asks how brain mechanisms underlie these phenomena. But I'm going to step back from all of that for a moment to start off with here, and just start at a really sort of simple starting point, which is we all have brains. This is a brain. It's actually my brain, but yours would look very, very similar. This is a brain's view from above. In order to interact with the world, you have to get the stuff that's outside of your brain into the brain, right? Because the brain lives in a box, it's in your skull. And the only way you can get information in is through things like your sensory receptor organs, so your eyes, your ears, which is what we're going to focus on today, but also your sense of touch, smell, taste. These are how information about the environment gets inside of this box, which is in which your brain lives. And the textbook view of how the brain works is that you have information that comes in. Can you see my pointer online? Let me just, does it work? Okay, fine. So you have eyes, you have ears, you have an auditory nerve, you have an optic nerve. Information from these sensory systems goes in and goes to kind of dedicated bits of the brain. So at the back of your brain, marked in red here, we have the visual cortex, and tucked in the side of your brain, marked in blue here, along the temporal lobes, just behind your ear, is the auditory cortex. And the idea that you will still find in neuroscience textbooks today is that these dedicated centers exist to process information from each of these modalities, and then this information is put together somewhere near the front of the brain in kind of more clever sort of cognitive parts of the brain, the frontal cortex, parietal cortex, places like that. And what I want to sort of do today really is kind of challenge this as an idea and leave you with the more realistic impression, which is that there's actually crosstalk at really almost every level of these sensory pathways. So how do we know that? Well, if we look inside these brain areas, so like within each brain area, you have hundreds of thousands or even billions, millions of neurons, of nerve cells. We can stain them and they look like this. They're very pretty. We can poke electrodes in here and we can record the electrical signals that they make to communicate with one another, and we can understand something about what sort of information they might be encoding. And if you do that in primary auditory cortex, so if you poke an electrode in, you record these electrical signals, and neurons communicate in what we call action potentials, little impulses, and we can essentially take the timing of these action potentials to understand what a neuron's doing. And as neuroscientists, and this is about as complicated as a neuroscientist is going to get today, we like to plot these as what we call raster diagrams. So in this experiment, we had an auditory stimulus, which was a 100 millisecond noise burst, a visual stimulus that was just a 100 millisecond light flash, and an audiovisual stimulus, which was the two presented together, so really simple stimuli. And we presented them about 50 times each in a random order, and then plotted as little dots each time the neuron we're recording from fired one of these impulses. So you can see by the fact that you get this kind of barcode-like look, this increase in spike rate here, this neuron responds to sounds, and it responds to a stimulus, an audiovisual stimulus, in very much the same way that it responds to sound. This is what we would expect to see in auditory cortex. It's a neuron that's kind of doing what it says on the tin. But we also found neurons that look like this. So here, the neuron indeed responds to sound, but it responds weakly to visual stimuli. and really quite strongly when we put the two together. And then we find neurons like this third one, which frankly looks like it got lost. It should be hanging out in visual cortex because it only really responds to visual stimuli. And even more curiously, we have neurons that really seem to want the combination of a sound and light together in order to elicit a response. And as I say, these are in what we call primary auditory cortex, so really early on in the auditory pathway. And so something that's driven the research in my lab for kind of more or less 20 years now is trying to understand what the point of this is. Like, why should there be neurons in auditory cortex that are responding to light? And we can do things like ask how many neurons are there, and we see that as we go from sort of early to secondary to kind of higher auditory fields, the proportion of neurons that either respond to light or have their response to sound modulated by light increases. We can kind of also do some anatomy. Again, don't worry about the details of this. It's just to say that there are really quite specific projections from different bits of the visual pathway to different bits of the auditory pathway. And again, that sort of suggests to us these projections might play a role. So I'm going to step back now and ask a more simple question, which is, when does seeing and hearing interact? And the spoiler alert is really all of the time. How do we know this? Well, one way that we know this is through sensory illusions. So a really, I guess, like old illusion at this point is the ventriloquist illusion. So this is the idea that you have a ventriloquist who has a dummy, and they move the dummy's mouth in time with their own voice while keeping their own face very still. This is so-called throwing your voice. And you can get this really kind of compelling illusion that it's the dummy itself that's speaking and not the ventriloquist. A much more kind of modern day example of this is every time you go to the cinema or the theatre and you're watching someone on a screen in front of you or on a stage in front of you, and you have the perception that their voice is coming from their mouth, when in fact, if you close your eyes, you're often aware it's coming from speakers that are all around you in the auditorium. So this ability, particularly of faces, to capture the location of a voice is really strong, and we're very often not aware that it's happening to us. So this is an example where really vision is dominating. So you see where you think the sound source comes from, and that's where your brain decides it comes from. But we have kind of corresponding illusions where really it's hearing that wins. So there's an illusion called the flash beep illusion, which I'm not going to try and show you because it definitely wouldn't work on Zoom. It won't even work on the room. But if you're interested in it, Google it, and it works very nicely in your own web browser. So this is an illusion where essentially you ask someone in an experiment to kind of fixate on this white cross and then to count the number of flashes that occur. And it's very simple. It's not like complex maths. There's like 1 flash or 2 flash, or sometimes maybe like 3 flashes. And on some trials, you pair the visual flash with sounds, so short beeps. And as you'd imagine, if you pair 1 flash with 1 beep or 2 flashes with 2 beeps, you'll perceive 1 or 2 correspondingly. Things get more interesting in the kind of, the labels have gone, conflict condition, where in this case here, where you have a single flash and 2 beeps, you'll perceive there to be 2 flashes. And in fact, if you do EEG over someone's visual cortex, you'll see 2 bumps, one that corresponds to the real flash and one that corresponds to the kind of flash that their brain just invented. And similarly, if you have 2 flashes and only 1 sound, your brain will fuse these 2 flashes together and you'll perceive there to be just 1. So here, it seems like the the fact that you can process sounds with a higher temporal accuracy is meaning that when faced with this conflict, your brain kind of opts to believe what it hears rather than what it sees. I'm going to show you one last illusion. I hope that it will work on Zoom, but if it doesn't, this is a BBC clip that's available, so you can kind of play around with it for yourself. What I'm going to do is I'm going to play it, and then I'm going to ask at least the people who are in the audience physically here to just tell me what they think he said. And as you heard a little snippet of there, it's not very complicated. So let's try that.
Ba, 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 ba.
Any takers on what he said? Ba, B-A, yep. Very basic. That's enough of that one, thank you. So we're going to play the same game again with a different clip.
Ba, 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 ba.
So any takers for what he said? What he said that time? So who thought it was far, F-A? Quite a few people. Who thought it was var, V-A? Yeah, some people. And who thought it was bar, because you know that I'm tricking you. So I'm going to play it through again, and to make sure, because, you know, obviously I'm an untrustworthy neuroscientist, to make sure that I haven't tricked you, watch it through once, and then just close your eyes and experience the change in perception that you get.
Ba, 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 ba.
So hopefully you'll find that when you close your eyes, you heard the audio that was unchanged all along, which was bar, B-A. The mouth movement he's making is far, F-A, and then depending on all sorts of factors like where you grew up, what language you speak, how good the projector is, how far away you sit from it, whether you've got your glasses on, whether you need your hearing aids adjusted, you'll either hear something that ranges from bar through var to far. Most people hear far or var with this thing. And even being aware of it really makes it quite, even when you're aware of what's going on, it's very difficult to break. So I've used this clip, you know, hundreds of times in undergraduate lectures, in talks, whatever. If I look at his face versus when I look away from his face, I hear different things. So I think it gives us an insight into how obligatory this process of linking what you see and what you hear is. So these, oh no, quite enough of him. So these sorts of illusions have led to the idea essentially of the brain as being some kind of Bayesian integration device. So we know that vision is a relatively slow sense. So it takes time for light to travel to the back of your eye, to hit the photoreceptors, which go through a kind of chemical signaling cascade to eventually elicit a neural response that then goes off to your brain. In contrast, in hearing, everything is very rapid. So we have hair cells that sit in the ear. They're physically gated by the sound pressure wave. So the rate at which an action potential fired is much faster and has a much greater temporal acuity to it. In contrast, your auditory spatial resolution is quite good. So it's maybe like 1 degree, which is sort of your thumb at arm's length. But your visual spatial resolution is a good 20 times better than that if you're foveating, if you're looking directly at the thing you're trying to resolve the position of. So the idea is that in this sort of Bayesian framework is that your brain weights signals according to how reliable they are. So if you're trying to work out where something comes from and you've got an estimate based on vision, which in this case is quite poor, so maybe it's like a foggy day or something, whereas your auditory estimate, which is the green line here, is really quite sharp, then the statistically optimal way of integrating these signals is to weight the more accurate, the less variable sense, more strongly. And you can play around with this. So now if you make vision more, to have a higher acuity, less variance, then you weight the visual signal more strongly. And the optimal thing to do if you have two signals that are have equal variance to them is to come up with something that lies between the three. So here, essentially, you have something that's a bit like the ventriloquist illusion on the right, something that's a bit like the flash beep illusion on the left, where audition dominates, and something like the McGurk illusion, which was the face and voice that I just showed you in the middle, where you have two estimates which are both judged to be actually quite faithful signals, so you kind of put them together. So another way of looking at this is that one of the reasons to integrate information across sensory modalities is to resolve ambiguity. So sensory signals are always noisy and often ambiguous. So essentially, by having two estimates of a signal, often actually that are, will contain some redundant information, but because, for example, vision and hearing are qualitatively different, you'll get some non-redundant information as well. That allows you to make sense of kind of a noisy and sometimes confusing world. And I'll just give you one more sort of demo for sort of how vision and hearing interact. So I'm going to ask you, I'm going to play this little movie in a minute. I'm going to ask you to tell me, or to kind of decide for yourself, whether these two green balls pass through one another or whether they bounce off one another. Okay, you can stop. Put your hand up if you thought they passed through one another. That's almost everyone. Put your hand up if you thought they bounced off one another. Okay, so as most people thought it passed through. Let's look at another example. So here, and again, this is where I'm afraid if you're watching this on Zoom, if it's messed with the audio and visual latencies, you may not get to enjoy this particular pleasure. But hopefully, I think the people in the room at least will agree, in the second case, you got quite a strong perception that the balls repel each other rather than passing through each other. So in this case, the visual image is completely ambiguous, right? There's no information with which you can faithfully judge whether these two balls are passing each other or bouncing off one another. So simply by adding in a sound at a precise moment in time, you can change your perception because you just add some more evidence in favor of one theory over the other. Cool. So I'm going to sort of step back away from sensory illusions and back to this question of, okay, so if we're thinking about signals that either help you resolve ambiguity or that are weighted in a kind of Bayesian sense, these are quite sort of high level, I would say, processes in which you would hope to have sort of undiluted or unpolluted veridical estimates of what you saw and what you heard so that you can then make some decision about how to put them together. But what we've seen in the lab and many other people have seen in all sorts of different species is that there is, at this really early stage, as I said, this crosstalk between the sensory systems. So I'm going to spend sort of the later part of this talk telling you a little bit about some of the things we're doing in the lab to try and understand what, in particular, these early sensory connections might be good for. That's what I just said. So essentially, yeah, we're drawing this white arrow on here to kind of recognize that these areas are probably not blue and red, but more like sort of different shades of purple. Okay, so one example, as we just saw with the McGurk effect, in which seeing the thing you're trying to listen to is really helpful, is lip reading. And we've all experienced what it's like to not be able to lip read over the past few years, and I think have a sort of now an intuitive impression of how much lip reading can help you when you're in a noisy environment. Now, obviously, when you see someone's mouth movements, that provides you with a degree of phonetic information. So again, we saw that with the McGurk effect. Some particular sounds like far versus bar and other consonants have really different mouth movements associated with them. That means that when you're watching a talker's face, even if you can't hear their voice at all, you can make some estimate of what they're saying. But a question that we've been interested in the lab for a number of years now is there a role for vision in this more kind of basic auditory process of actually sorting out a sound scene in the first place? So this idea that, you know, all of the sounds in this pub are going to arrive at your ear in this mixture, and that what your brain has to do is separate out the voice of the barmaid from the music in the background, from your friend who's next to you, so that then you can kind of take the next step and say, okay, actually it's the barmaid that I want to concentrate on listening to, or it's my friend I want to listen to, and actually choose a source to pay attention to. And one idea that we've had is that in addition to lip reading providing you with these kind of phonetic cues, it potentially provides you with sort of rhythmic cues. So as your mouth opens and closes in a way that tightly correlates with the amplitude of the sound wave that's coming out of your mouth, you also tend to make rhythmic gestures as you speak. There's a whole variety of quite low level, I would say, language independent cues. And you can also think about other things, like if you're watching a band on stage and you watch the guitarist's strumming motion, it's much easier to pull the guitarist out, or you're listening to an orchestra. If you want to hear what the cellos are doing, looking at the cellos really helps because their bowing motion gives you some sort of extra information that allows you to pull the correlated sound sources out of that mixture. So we've been really trying to look to see whether these early cross-sensory connections might support this sort of lower level integration that's really helpful to us, and not just to us, really to any animal that has to make sense of an audiovisual scene. And we've been going about that a few different ways. So we've been doing some experiments with humans. So we kind of create a sort of impoverished version of this Friday night pub scene here by sticking people in a quiet room and playing them sentences which have kind of very little predictive structure to them. And essentially they're told they have to listen for a target sentence, which is show the dog where a colour and number is. We make it a little bit more difficult by at the same time having some competing talkers who say things like show the sheep where the green clock is, or show the cat where the red bowl is. And to understand the role of vision, we do a couple of different things as well. So we can either present these sounds with just a still image of the talker who said them, or we can present the sounds with a video that matches the target talker, or a video that matches the masker talker, so one of these other sentences. And we can ask how these different visual conditions affect people's ability to accurately report this colour and number. But to try and get at what kind of extra cues you might get that are not lip reading, we also have a condition that's a bit like being in a really bad Zoom call. So you have the target talker saying the words, but when you get to the actual keywords that the person has to report for the task, the video freezes. So just for that kind of fraction of a second, when the information about the mouth movements, or the mouth movements actually would allow you to kind of hear what the target words are, we freeze the video and it resumes again for the last word of the sentence. So in this case, any visual benefit can't be because the person's lip reading the actual target words. So what happens? Well, in young normal hearing listeners, if we contrast the threshold that you measure, so essentially what we do is we vary the sound level of the distractor talkers and the sound level of the target talker, and we find the threshold at which it's sort of now significantly difficult for you to understand, or you only report sort of 70% of the time the correct colour and noun, and that we call that a threshold, and we do that repeatedly, and then we compare the thresholds across different conditions. And we can then measure something that we call a visual benefit, which is how much better, so how much quieter can you make the target sound and still understand what's said when you have the video of the target. So here we have 30-something normal hearing listeners, and you can see that almost all the listeners have a kind of a positive benefit. So they were essentially better able to follow speech when it was very quiet, when they had the talker's face in front of them. And that benefit persisted, albeit obviously it's smaller, in this what we call the interrupted condition, so where they couldn't use lip reading. So that suggests to us that there are some additional cues that people are able to use to actually follow the target talker through the mixture, so that when the actual keywords come about, they can still benefit, they still show a benefit relative to a sound alone condition. We've tested older listeners who generally show smaller benefits, in people with hearing loss who seem to have perhaps slightly better ability to make use of the uninterrupted condition, and sort of a reasonably similar proportion of people show some benefit, again not a very impressive benefit in this interrupted condition. And we've done some modelling to kind of tease apart with this 125 listeners what things affect your ability to benefit from seeing a talker's face. Some of the things that are sort of interesting and repeat things in the literature is that if you're a female, you're much better at making sense of benefiting from seeing a talker's face. For whatever reason, men seem to benefit less from visualising a talker. Hearing loss didn't have any effect. Age didn't really have any effect either. And your ability to lip read in silence was not as predictive as your ability to benefit from this interrupted condition. So I think what this kind of slightly complicated statistics is showing us is that there are potentially two types of audiovisual benefit here that we can pull out. There's something that comes potentially from your ability to separate the target talker from the background talkers, which would fit with this sort of low-level integration idea, and then lip reading on top of that. So we've been wanting to take this sort of one step further, really, which is to take language out of the equation altogether. So we know that lip reading is a thing, right? People have reported that for decades. It's not terribly surprising. So can we actually go one step further to saying that one role for visual inputs into auditory cortex, or one role for integrating vision into hearing, is to actually solve this challenge of auditory scene analysis? So to do this, and this is work that we did with Ross Maddox, who's now at the University of Michigan, Ross actually designed this stimulus set that was essentially an auditory selective attention task. So on every trial, people heard two sounds. One sound started a second earlier than the other, and that was your cue that that was your target sound. And both of the sounds fluctuated in amplitude at roughly the rate that syllables are produced within a sentence, so at about 7 hertz, but as you can see in a sort of noisy rather than periodic way. And every now and then in these quite long trials, so the trials were 14 seconds long, there would be a little either pitch or timbre deviant, and people had to just press a button when they heard this little pitch wobble or this little brief change in the timbre of a broadband sound. And they were to report those changes only if they occurred in the target stream and suppress their urge to press a button if they occurred in the masker stream. They also saw a visual stimulus. I'm going to let you experience just a moment of the joy that the listeners experienced in performing this quite long experiment by letting you experience those stimuli here, because it's just easier for you to experience them than for me to say them. So your job is to keep looking at this white dot here. You're going to get 1 second at the beginning where there's only the target sound. So your job is to latch onto that target sound, and then you can kind of mentally try and press a button when you hear these little pitch changes in the target stream. And I will spare you any more of them. So if you pressed the button anywhere in kind of the first 2/3 of the trial, then you pressed it for the masker stream, and that was not good. If you pressed it somewhere towards the end of the trial, then you correctly detected the target. So for each person, we kind of adjusted the difficulty so that this was quite a difficult task. I don't know if you noticed anything about the way in which the visual stimulus was changing. So it was actually changing in a way that was yoked to the amplitude of the target stream. So every time the target stream got louder, the visual disc got larger. Every time the target stream got quieter, it got smaller. And that was this visual condition here, so a target coherent condition. We also had a condition where the disc changed in time with the sound that you were trying to ignore. And we had a third condition where the disc just did its own thing. It changed independently of either stream. And what we found, it's probably best seen looking at this panel on the right here, was that in this, and I'm sorry, the target label seems to have escaped somewhere along the way, people were better able to detect these little pitch deviants when the visual stream changed in time with the target stream than they were when it changed in time with the masker stream. And the independent condition was somewhere between the two. So it seems like you get a benefit when it's target coherent. You actually get an impairment when it's masker coherent. And it's important to remember with these stimuli that there's absolutely nothing in the visual stimulus that tells you about when these little pitch perturbations were in the sound. So if you took your headphones off to do this task, then you would not be able to do it at all. So this is quite different from something like lip reading, for example, where actually quite often you could have a fair stab at what the word might have been. You might have been able to see how many syllables there were or what the starting consonant was. Here the visual information to solve the task is completely lacking. So the only thing it can do is either allow you to better separate these two sounds and or allow you to better focus on one of them. Like maybe the audiovisual stimulus is just better at attracting your attention, and that would help you when it's target coherent and hinder you when it's masker coherent. We actually think it's the first. So we think that this visual stimulus is allowing you to pull these sounds together more effectively. And the reason for that is because when we go back to recording neurons in the brain, again, I'm going to show you some of these raster plots, we see single neuron correlates of this human behavior. So we can present these stimuli either on their own as a single audiovisual stream, which is shown up here in red, and again we get this pattern of spiking underneath. We have a different audiovisual stimulus here shown in blue. It elicits a different pattern of spikes. We can use a decoding approach to take any one of these trials and accurately say which stimulus elicited them. But the interesting part of the experiment comes when we present these two streams at the same time. And now, depending on the properties of the neuron, you might predict that it might respond to one sound source over the other if one was sort of one that better fit its receptive field, or you might predict that it might respond every time the kind of cumulative sound wave got loud. But what we actually see is that we can flip the neuron from responding. So now we're going to present this mixture of sounds over and over again, and we're going to separate the sounds trials out into trials where the visual stimulus changed in time with the blue stream or changed in time with the red stream. And here we can effectively flip this single neuron from responding to one sound in the mixture to responding to the other sound in the mixture, simply by pairing it with a visual stimulus that matches its temporal dynamics. And that's underneath the responses to the two streams on their own to show you how similar the response to the mixture is to each one of those sounds on its own when you give it the right visual stimulus. I don't think we need that. And we can even see correlates of this ability to detect deviance that we see in the human listeners. So here we have two types of trials where you have exactly the same sound. It either has one of these little pitch perturbations or timbre perturbations in it, or it doesn't, which is the red and the black. You can see by the bumps in this neural response here, this single neuron represents these deviants when they're present, but that response is much less when it's accompanied by a visual stimulus that's changing independently of it. So to sort of summarize what I've told you and what maybe you might want to come away from this with, auditory visual integration is really something that's happening all of the time, whether you like it or not. It's kind of an obligatory way in which your brain works. We can reveal the nature of these interactions through things like sensory illusions, which of course you rarely face in the real world. Like it's very, the ventriloquist and his dummy is a rare kind of talented exception who can separate out, physical sort of stimuli that come from the same object. There are multiple points in the brain where auditory and visual signals converge, and I think it's sensible to imagine that depending on the point at which they are brought together, there might be different roles for integration. And certainly something that we sort of try and argue at the moment in the lab is that one of the roles for these really early integration of sensory cues might be to support, particularly for hearing, might be to support this challenge of separating out sounds in a mixture. I'd like to thank the people in the lab, the people who particularly contributed to this work. It was Stephen and Hurrier with the electrophysiology, as well as Rebecca, and the human behavioral work was both leaders and some work that we did with Adrian Lee and Ross Maddox in the States. And I'm happy to take any questions.
Should we take one question in person and one online? Do we have a question from here?
Sure, thanks. It was extremely interesting. Would you be able to go back to the slide early on where you were showing the response of the auditory neurons to visual stimuli?
That one.
So what interested me here was B, where you see, I guess, if I understand right, that is a response of an auditory neuron to a purely visual stimulus, and yet it appears delayed in some way. And of course, we know that there's a much greater latency in the visual system than the auditory one. So could you talk about, does that tell us anything about kind of where that connection takes place or how, and what are the implications of this kind of cross-modal coupling, but apparently with a latency that comes from the visual system?
Yeah, so I mean the latency, I guess you have sort of two sets of latencies, right? You have the latencies of the physical stimuli in which light travels faster than sound, and you have latencies within the brain in which visual signals are delayed.
Right.
And we can use those latencies to say something. So for a ferret visual cortex response, for a ferret visual response, the timing of this one is about what you'd expect for V1, for example. So it tells us that potentially it's cortical, but it's probably not feedback from a higher area. We've done some inactivation experiments where we silence visual cortex, and some of the later responses go away and some earlier ones persist. So my intuition is that actually there's some crosstalk happening earlier than auditory cortex in the thalamus or colliculus. And how the brain deals with these differences in latency, I think, is a really interesting question. I mean, I guess as you move up the hierarchy, the window over which neurons integrates gets broader, and the less a kind of 20 millisecond, 30 millisecond difference becomes potentially a problem. And we know that from behavioral work, you can adapt to constant offsets, right? Like if you give someone a load of conditioning stimuli with a 200 millisecond lag, your perception of when things occur at the same time shifts to compensate for that. So that too must be happening somewhere in the brain, but possibly not this early on. But yeah, the differences in latency is a really interesting thing, especially for the experiments with the temporal coherence where the physical stimuli are changing synchronously. So whatever it is potentially entrainment, or yeah, I think the absolute mechanism is still up for grabs.
Thanks. We have a question online from Edward Bale. Can you comment about the cocktail party effect where a known sound can be heard above other?
A known sound, like I don't know what I'm asking. I guess that's an attentional effect. So if you hear your name or something like that. So I guess there are some really nice studies from recordings from human auditory cortex that show if you present someone with a mixture of two voices and you ask them to pay attention to one voice over the other, that by the time you get to higher cortical areas, it's almost exclusively the voice that you're trying to listen to that is represented. However, still in the kind of early sensory cortex, both voices are present. So even when you're paying attention to one source in a mixture, the other information is getting in and is probably being processed in some level. And I guess certain sounds that attract your attention very strongly, like your name or something like that, can then direct attention back to make you switch what you're kind of preferentially representing. And I would imagine that something that also makes you turn and look would actually give like an added boost to that. But hopefully that answered your question.
Another question from the audience?
Maybe just not sure how it relates to the neuroscientists, but we as kind of design engineers want to know the implications kind of for design. Do you look at this as well? Okay, we know all the things, now how do we translate to make interventions?
Yeah, so the sort of the next steps for the human work that we're doing is to try and understand, in terms of interventions, is to try and understand like if there are more than one way in which visual inputs can help you to listen. Essentially, like if you come to me and you have a hearing loss, I'm going to give you a set of hearing aids, but maybe I should also test your lip reading and your basic auditory visual integration to say, hey, you should go to CityLit and do a lip reading course, but actually you should take this iPad training app, and actually we should train you to be able to integrate your signals better. Because we've also done some other experiments where, yeah, like if you train people to detect these temporal coherence relationships, they get better at using them. So that's sort of the way in which we think about having some kind of further like impact of this. In terms of sort of design in like a bigger picture of design of spaces or things like that, yeah, I'm not sure. I'll ask Lorenzo. More his cup of tea.
Thank you. A question online: how does vision loss affect the data, since those that have low vision or blindness may not receive visual cues?
Yeah, so we know from animal work that, for example, and it's all quite sort of sensory system and or species specific, but for example, like if you make a normal hearing ferret deaf, somatosensory inputs take over auditory cortex very quickly. So I think a lot will depend on the nature of the loss and how long term it is. I think if you have a visual loss, if your visual perception is less acute, then you will rely more on your hearing, and you will integrate things. Your Bayesian brain will integrate hearing more strongly than vision when there's a conflict, and these things are kind of adaptive. Whether people who are blind from birth presumably don't benefit in the same way in an auditory scene analysis situation. But yeah, slightly fluffy answer.
Unless there is someone else. I have one, but I can wait. No, you're one.
I wanted to pick at the latency a little bit more, and just in the illusion with the ba, fa, fa, this sort of post hoc, somebody you have this stimulus, and then you say, what did you think you just heard? Then I guess there's quite a time window in which you can kind of reflect and integrate on those two stimuli. But has anyone tried to sort of unpick kind of in the immediate moment in some way, kind of how that perception unfolds over time?
So I don't know if they have in exactly the way you mean, but one thing to kind of bear in mind is that mouth movements lead the sound of your voice by 200-300 milliseconds. So in the real world, for speech, actually, like, mouth cues are tremendously predictive of the sound you will hear in 200 milliseconds time. So that actually begins to give you some kind of compensation for the latencies within the brain, right? Because if the visual signal is effectively arriving 200 milliseconds earlier.
Yeah, interesting. Yeah, and then just to contextualize this, I mean, I come at this as a musician and somebody who works in design of musical systems, so I'm always very interested in kind of what can we say in the moment as opposed to kind of looking back on it. And so I guess I'm just curious about what kind of studies may have been done in terms of looking at audiovisual integration. In some, I mean, I don't know if there's fMRI studies or anything that can be done to look at exactly at this moment, this is what the brain is doing, as opposed to asking somebody after the fact, what do you think you heard?
Yeah, so I mean, there are lots of fMRI studies that try and pin down different elements of a multisensory computation on different bits of the brain. It's very hard to do anything with a resolution of less than a couple of seconds with MRI. So it tends to be the sort of causal inference happens in kind of higher areas of the brain. So that's where you make your kind of slow trade-offs of like, did I think it was one source or two? And therefore, how should I integrate it? Whereas these sort of more, the sensory areas do show multisensory integration, but it's much more temporal and spatial coherence driven. So I think there are different computations at different points in time. I think it's really hard to get at them with a lot of the tools that we have at the moment in a human. Although, increasingly people are recording from human brains of epilepsy patients, so maybe we'll get somewhere along those. Because as soon as you ask someone, so you can do like a speeded reaction time judgment, but that's still not a kind of great way of getting at exactly what they perceived, because then you need quite a limited number of options.
What about like the EEG on board, where like you just see if they detect a difference and then present the visual stuff?
Yeah, I mean EEG would be a better way of doing it, because obviously you get back time.
You wouldn't know what they exactly heard, you know that different.
There is a quick technical question from Jonathan Higby. How was the audio presented, the experiment shown? Earphones or loudspeakers? If loudspeakers, were they directly in front of the participant?
So the animal work was a mixture of speakers, like headphones and free field. The human work was all over headphones without any kind of particular spatial cues. So they would have perceptually appeared from kind of within the center of the person's head, effectively.
I have a random question. I remember reading a nice story from the Oliver Sacks and his book Musicophilia, where I think it was one of the beginning. It was about himself that came back home after a long day, put on a Mahler symphony on the vinyl and sat down and listened to it. And when he finished, he realized that he never switched on the vinyl player. I was just wondering if you had carried out an EEG during or any sort of experiment that you do to measure something about the brain, do you think you would have seen something related to the inexistent?
I'm pretty sure people have done that, yeah. Like if you imagine speech, or if you imagine music, or you imagine it's even like the locked-in coma patients, right, who imagine playing tennis as opposed to imagine listening to music, and you get different neural signatures of those two things. So I think, yeah, a lot of, like if you're hallucinating music, a lot of your auditory cortex will be reconstructing the music, would be my guess. I haven't worked out how to train ferrets to do that yet, so.
And another, can I see, I think it was a little bit later in the slides where you show both the auditory and visual envelope, I don't know what it is.
I think I know what you mean. Somewhere around here, like.
Oh no, it was another one where it was earlier, I think. No, earlier. It wasn't really the envelope. I call it envelope because.
Take a pick. Earlier. No, we're at the beginning of the talk now.
The one where you showed the diagram where it says, when it shows which one takes over, but this one.
This one. Okay.
So what is position? Because I'm not 100% sure.
Oh, I mean, it's a cartoon, but I think here it's sort of supposed to be potentially like an estimate of spatial position.
Okay.
But that axis could be anything, right? It could be size, it could be shape. It's like, it's some perceptual judgment, and the idea is that you get one estimate with one sensory system, which has a mean and variance associated with it, and another estimate from the other sensory system with a potentially different mean and variance.
Because, for example, the example of the reliable visual and poor auditory, I'd argue that the McGurk effect, the audio is not poor, it's actually more informational than the visual.
Well, yeah,
Except
I mean,
the visual takes over.
but the vision is so reduced, isn't it, compared to, it's a single syllable. So I guess like for that single syllable, the visual cue is quite unambiguous. But it depends on who you are as a person. Like
But
different
I'm
people have
thinking,
different
for example,
functions.
From a machine perspective, I would find it much easier to detect what the person is saying from the audio rather than from the visual. The visual would be more complex. So to a certain, and also we're talking about communication for which we use mainly auditory stimuli, not visual. So I'd say, why is vision taking over there? It's annoying.
Could it be because of like multi-talker environments? Like usually when you hear speech, there's like other things going on, so the auditory signal's not like on its own. So like having lip reading abilities is very useful.
And I'm wondering, is this maybe going to change in the future if we talk more and more on the phone without seeing people? Would we get more and more used to just trust our hearing without?
Yeah, I mean, the truth is, right, like the McGurk effect that never happens in real life. Like there's no way it would be physically impossible for you to generate that stimulus in the real world. Like you can generate it digitally, but so your brain is completely overwhelmed by instances in which the two say the same thing. So in this very artificial situation where we create this conflict, and like, you know, primates are incredibly visual animals. Like we really rely a lot on our visual system. So my guess is it's just one of those little quirks of.
You talked about cocktail party effect and things like that, but they're kind of all relatively new things on a human timescale. I'm talking evolution. What sort of benefit does this audiovisual cross interaction have from an evolutionary point of view? I'm thinking in terms of finding dinner and spotting things which are trying to treat us as dinner.
Yeah, so I mean, first of all, I don't think cocktail parties are a particularly uniquely human phenomena. Like frogs have cocktail parties and use many of the same cues that we do to find their mates in the kind of cacophony of many others. And there's lots of species who have to discriminate an individual from a crowd of others. So I think there probably are lots of things that are speech specific in humans, but lots of things that are not. But if you are trying to catch your dinner, it's very useful to you to integrate that tiny rustling noise you just heard with that tiny like flash of movement you saw. So I think the idea that putting together quite, you know, poor and sparse is maybe the right word for it, sparse signals that prey may be emitting, something like Dan Goodman here has been working on, actually, you should talk to him about it, is like a very good example of something that's incredibly adaptive for an animal to do, right? Like if you're prey, you maybe don't want to wait until you can smell, see, touch, and hear the cat because it's too late. But like if you have a slightly noisy signal, then combining the tiny bit of visual information you have with the tiny bit of hearing or olfactory information you have is pretty adaptive, I think. So in that sense, I think like transient signals that are locked in time together get kind of a preferential boost through the system, which you can then bootstrap onto to kind of get a benefit in a cocktail party, but I think probably comes from a much more low level kind of integration of like ambiguous or sparse information.
We have a question online from Alexandra Alvarez. Did all participants listen to the audios at the same dB level? How did you determine the hearing spectrum of the participant? Because everyone does not have the same audible range.
Yeah, so for the sort of speechy experiment we did, well actually for both sets of experiments we did audiograms, so we knew exactly what their hearing level was, and then we used that actually to try and understand whether that affected their kind of benefit of a visual stimulus. For the non-speech experiments, we ran a little sort of threshold test beforehand on everyone to actually pick a difficulty level that meant that each participant found the task equally difficult, which did not mean that they ran it at equal sound levels. So some people would have run it at a harder, in absolute terms, level than another, because we were interested in finding this level of performance where they're not getting every trial right and they're not getting no trials right, so you've got kind of room to move in both directions. Yeah.
Is there one last question from here? Yes?
So with the headphone source, have you seen a difference, or have you tried a difference between out of phase to produce a diffuse sound as opposed to in phase, which would be highly localized?
Yeah, I mean, so we haven't done that exactly, but we have done quite a lot of stuff with sounds that vary in their kind of real world spatial position using speaker arrays and stuff. And a lot of these multisensory effects are really quite, well, the ones that are based on kind of timing are really quite robust to spatial differences between the audio and the visual signal. I think because in that case the visual signal's kind of spatial position just dominates.
I'm looking at the slide you have up now and wondering how the prioritization might change between in-phase signal and an out-of-phase signal.
Yeah, so I think.
Or other things being
Yeah, so no, I think a more diffuse signal, if it was a spatial judgment, a more diffuse signal would get less weight. So there was Alaise and Burr did an experiment which was sort of dubbed the reverse ventriloquism experiment, where they took people into the Sydney Opera House and projected. It was the only place they could find to project big enough, blurry enough visual images. So they made the visual scene much more ambiguous than it normally is and much more low resolution than it normally is. And then people began to rely much more on their sense of hearing. So certainly, yeah, like the more diffuse the signal is, the less well you can resolve exactly where it's coming from, the more scope there is to be biased by some other modality, essentially, I think.
I missed an online question from Bill Milgram. Have you studied the brains of those with synesthesia to compare them to the other data you have collected?
No, I haven't, but it would be great fun. I mean, other people, yeah, plenty of people study synesthetes. I guess the challenge is finding enough of them to get them into the lab so like during COVID times we did lots of online experiments, and I think the only thing we demonstrated other than spending an awful lot of money was that running online audiovisual experiments is impossible for anything other than speech, because you can't control the latency mismatch of like running experiments, which is a real shame because it would be much easier to recruit synesthetes kind of online, I suspect, than around. But yeah, they're a really interesting subpopulation, and I think they're probably just one extreme of normal. So there's reasonably good evidence they have slightly stronger crossed connections than the rest of us, but we all have them. So.
Okay, if there are no more questions, there is a statement from Jonathan Digby that I read before the end, which is, ears are the eyes and the back of the head.
Wise words.
So thank you very much, Jenny, again. It was lovely, and thanks to everyone for contributing with questions, and hopefully I will see many of you at the next meeting. Thank you.